Welcome to this recording of the Greater Delaware Valley Chapter's 2011 MS Research Update. The first speaker is Dr. Ellen Mowry of Johns Hopkins University, discussing her work studying the potential of vitamin D to slow MS progression. The next speaker is Dr. Judith Greenspan of the Children's Hospital of Philadelphia, talking about her work to restore nerve function in people living with MS. The final presenter is Dr. Simon Gregory of Duke University, who discusses the genetic component of MS. The discussion is moderated by Dr. Clyde Markowitz from the Hospital of the University of Pennsylvania.
Vitamin D insufficiency does appear to increase the risk of developing MS. So people say, well, can vitamin D supplementation prevent MS? We don't know. It's a pretty hard study to do because it's likely that the risk period could start as early as childhood or even maybe in utero. We don't know. And so if that's the case, at what point in life do we have to start supplementing people, and how do you carry out a research study that's large enough to answer this question? It's a difficult one. The second question is, should all patients with MS be taking vitamin D supplements? Because most of us don't go out in the sun and get enough sun, even in the summer. We all put on sunblock. And we're mostly not getting enough sun to keep our levels of vitamin D up these days. Well, there is some evidence from mice that have the mouse form of MS that vitamin D may actually reduce the severity of the disease. So mice who develop this model of MS, who are exposed afterwards to vitamin D, actually develop a less severe form of the disease. Interestingly, this was more true in female mice than in males. But what about humans? I'd say the jury is still out. We have some observational data as well that I'll show you, but we don't really know definitively that vitamin D supplementation will help. Why might it not be a good idea to just give everyone vitamin D? The graph on the left is actually a complex graph of the immune system, and you don't need to know the details of it. But vitamin D actually impacts many aspects of the immune system. And we don't know exactly what effect it has where, and we don't know if it impacts the immune system of people with MS the same way as it does the immune system of healthy people. Secondly, everybody's genetics are different. So for example, I was talking with the board earlier. There's a gene that has been associated with a high risk of MS, and vitamin D actually increases the activity of that gene in the lab. So does it do that in people with MS, in living people? And if so, would vitamin D actually be good for those people? I'm not sure. Third, there are some people who are at risk for developing complications of too much vitamin D. For example, it can increase the calcium in your blood or in your urine, predisposing you to kidney stones. So just saying everybody should take vitamin D may not be the smartest idea. But we do have some evidence that's supporting that vitamin D may be helpful for people with MS. One was in a study that I worked on at UCSF in San Francisco, looking at children with MS. And we saw that those children who had higher vitamin D levels were more likely to not have an attack over the following several months. And in fact, if a person had a higher vitamin D level by 10 nanograms per milliliter, which is just a concentration of vitamin D, they had a 1/3 reduction in the risk of subsequent attacks. A very similar type of effect was found in Australia in adults with MS. So I think these results are really encouraging. We also were interested in exploring if vitamin D was associated with the development of new spots, or lesions, as some people will say, on the MRI scan. And many of you may have seen MRIs in the past of MS, and both of these are pictures of the brain. And on the left, the arrows are pointing to MS spots, areas of inflammation in the brain. And the white spot you see on the right is a spot that is active because it's lighting up after contrast is given. So we were really interested in understanding if vitamin D levels were associated with the development of these spots. And so we actually did a study at UCSF of almost 500 people with MS. And we actually found that low vitamin D levels were associated with an increased risk of MS attacks as well. So each 10-unit increase or higher vitamin D level was associated with about a 15% lower risk of developing new spots overall, and about a 1/3 reduction in the risk of the active spots occurring. So after that study was published, again, people said, well, maybe we should be giving vitamin D to everyone. So I have to keep coming up with arguments why a trial is important. Here's one of the clinchers. Vitamin D is a supplement, and it's available over the counter. And some people say it's a vitamin. How could that be harmful? It's actually a hormone. And even if you did just say it's an over-the-counter supplement, we've had experience with several supplements and vitamins in the past that have looked really promising for other diseases, but then when they were studied in a more rigorous fashion, it actually looks like they were harmful. So, for example, folic acid was thought to be really good in preventing colon cancer in studies like those I've shown you for vitamin D and MS. When they were studied in a rigorous clinical trial, it looks like folic acid actually increased the risk of colon cancer. Similarly, beta-carotene, does anyone remember that craze? Was thought to be associated with lower risk of heart disease. And in the clinical trial setting, was shown to actually increase the risk of death from heart disease and increase the risk of death from lung cancer. So, I think we all know, as do animals, that sometimes you can have too much sunshine. Maybe you can have too much of the sunshine vitamin, or maybe there are certain people who shouldn't be taking as much vitamin D. That's why we thought it was really important to do this clinical trial, to make sure that vitamin D at least is not harmful for our patients with MS, and hopefully will prove that it's actually helpful. So the basic layout of the study is that people with relapsing remitting MS are being enrolled, and those who are eligible will be randomized to receiving higher doses of vitamin D, 5,000 international units a day, or to the standard of care, which is recommended by our Institute of Medicine, 600 international units per day. And everybody will take the same MS medicine, glatiramer acetate, or Copaxone. People say, how did you choose that dose of 5,000? And we basically looked at the research that was available and said, well, from the studies that were done in the military, it seems like having levels over, when we say the 100 nanomoles per liter, was protective against developing MS. And we saw the effect of vitamin D seem to extend up to 60 nanograms per milliliter, or 150 nanomoles per liter. So where those stars are is sort of the range that's marked out. And above that, we actually don't know if the benefit of vitamin D would theoretically continue, plateau, or even reverse course. So we wanted to use a dose of vitamin D that sort of got us in between that range. And you can see from this graph, which was a study of vitamin D supplementation, the amount of days that the study participants had taken vitamin D is plotted on the x-axis and vitamin D levels on the y-axis. And while people who were on placebo or received 1,000 international units a day had very little change in their vitamin D levels, 5,000 international units a day got them right in that target range that we had. And that's how we chose that dose for our clinical trial. So again, our goal for this study is to enroll patients with relapsing remitting MS. We calculated using mathematical formulas and what we already know about vitamin D and its possible effect on MS, that we need about 172 patients to definitely see if there was an effect of this high dose. And we're really looking to see if it impacts the number of attacks that people have over the course of the 2-year study. But we're also interested in exploring whether it reduces the number of new spots on the MRI scan, whether it's associated with quality of life, if it changes disability at all, and if it's safe. We have a very ambitious goal, which is to enroll all of our patients within 1 year, so that we'll have our results as soon as possible. So that's what we can use your help on. Refer your friends. There are a lot of sites already that are participating in this study, and we anticipate that there will be more. Here on the East Coast, we have a site at Mount Sinai in New York. Dr. Markowitz's group is participating in Philadelphia. I'm leading the group at Johns Hopkins. There's a site in Detroit, in St. Louis, San Francisco, Seattle, and Portland. So we're hoping to expand the number of sites, but that's where we're planning to start off.
You have to be on Copaxone, right?
So we'll get to questions at the end. So in conclusion, I think that vitamin D levels, we've shown, at least in these observational studies of people with MS, do tend to be associated with the amount of inflammatory activity, namely relapses and new spots on MRI. In people with MS, we still don't know if vitamin D supplementation is safe or effective, but this trial, sponsored by the National MS Society and really championed by this group, will really provide us with the critical information that we need to make decisions about how to advise our patients about vitamin D supplementation. Thank you so much.
Thank you, Ellen. So we're going to take questions after the other speakers are completed as well. So I just want to acknowledge that, this study is actually, when you raise money and you say, okay, I'm going to donate money to the MS Society, where's my money going? This study is actually fully funded from this chapter solely. So we are the actual responsible parties for this study. Okay, so you're going to talk down there, so that's okay. We're going to hear from Dr. Judith Greenspan, and she's going to talk about how to make MS better.
Okay, so can you all hear me?
Yes.
So I'm standing down here so I can see my own slides. That's why we fled the podium before, because we wanted to see the presentations too, and you can't see the slides from up there. So I have been an MS researcher since 1986, when my postdoctoral fellowship at Children's Hospital was funded by a grant from the MS Society, and I appreciate all the support that I've gotten from the MS Society over the years. What I'm going to tell you about today is the work that goes on in my basic science laboratory. It's very different than what you heard before, which I hope to make for an interesting contrast. I'll tell you a little bit about the aspect of MS that I work on, then the kinds of questions that we ask, and then a project that's going on in my lab that I think we hope will ultimately end up in some therapy. And in the meantime, you're going to have to learn some basic biology of myelin. MS is not a simple disease, and there are many aspects to it. And no one scientist can study all of MS. You have to have your area of expertise. You have to have what you're good at. So you could study causes of MS, not what I do. You could study the immune system. We heard a little bit about that before, also not what I do. You could study neuroprotection, which is a relatively new field of protecting the nervous system before the damage starts, which we heard a little bit about before. We're sort of getting warmer. But I work on repair. And to understand repair, you have to understand what myelin is. So, myelin is the membrane sheath that surrounds the nerves and lets the electrical impulses go. So you can think of it just like insulation on an electrical wire. So you all know that the electrical wires in your house, the copper wires, are covered with a coating. And if they're not covered by that coating, the electricity may not get from your wall socket to your lamp or your clock radio or whatever it is that you're trying to turn on, like the frayed wires here on the slide. So let's go back a step. Let's say that your brain needs to tell a part of your body, let's say your foot, to move. Your brain sends messages down the nerves. The nerve starts in your brain with a cell body, that's the little round thing at the top. And then there's a nerve process that goes down, 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 either to a muscle, and it causes the muscle to contract or relax, or to another nerve, which it then signals to, and then goes down a little further. So if you're trying to signal from your head to move your foot, you may need a number of connecting nerves for the impulse to travel down. Those nerve processes, which are called axons, have to be insulated, or it would take a year for that process to go all the way down it. I'm not going to use a lot of technical terminology, but a few words are necessary, and axon is one of them. So the axon has to be covered in a coating that allows the message to flow. So this coating is myelin, and myelin is a membrane, a living membrane that is wrapped around the axon and lets the information go down the axon. In MS, myelin is destroyed in places. Those white spots that you see in the MRI are areas where myelin is destroyed. And when myelin is destroyed, the impulse won't travel, and you can't move whatever it is that you want to move. So one way to think about myelin that I think is sort of effective is to think about a newborn baby. So a newborn baby is floppy and doesn't have a lot of control over its muscles and its body. But over the course of, let's say, 18 months, that baby learns to sit up, crawl, stand up, walk, and talk. A lot of the difference in the nervous system of that baby, a lot of the possibility to do this is myelin. As the baby myelinates, the baby gets control. Babies are born mostly without myelin. Myelin takes easily 20-25 years to be completed, but the first part of life is the most important. So if you want to think about myelin, think about that baby growing up and getting control of what it can do. So, let's see. I have a cartoon here that I'm going to use a number of times. So it starts with the cell body of a nerve cell, the neuron. Inside the little circles, a nucleus, and that nerve is extending an axon. That is the blue cell. The myelin is the pink stuff that's wrapped around the blue cell. Myelin occurs in little pieces called segments, and the message going from the nerve to the muscle goes along those segments. If the myelin is destroyed, as you can see on the right-hand side of the cartoon, there's either a distorted message or the message simply doesn't get there. That's the first problem with destroyed myelin. The second problem that's been recognized more recently, but is maybe even more significant, is that myelin supports the axon. So if myelin is destroyed, the nerve ending, the axon underneath it will ultimately, over time, fall apart. Then you have no myelin and you have no axon, so you have a double problem. So myelin has two functions. It keeps the nerve intact, and it provides insulation. So, why do I have a picture of Crisco up here? So what is myelin? Myelin is a lipid or a fat, and fat is white, like Crisco. So you all have heard of the white matter of your brain and the gray matter of your brain. Myelin is what makes the white matter of your brain white, because the white matter is full of myelin. So, myelin is made by cells called oligodendrocytes. I apologize for the name, but one way you can think about it is that oligo means many, like oligarch. And dendrite or dendrocyte is a process, like a tentacle. So oligodendrocytes are cells that send out many processes to contact the axon, the nerve process, and wrap the myelin sheath around it. So this is what myelin actually looks like. On the left-hand side, it's just a picture of a myelin segment, and imagine cutting a cross-section of it. In the middle, you have a picture where you have an axon in the middle. That's a sort of black and white spotted stuff. And then the myelin wraps around and around and around and around the axon, many, many, many layers, and it compacts to make the myelin membrane. In parts of your brain, like the corpus callosum, you have many, many myelinating axons, and that's the picture on the right. They're all packed in together. Okay, so during development, oligodendrocytes are generated from stem cells. Now, these are not necessarily the human embryonic stem cells that were controversial under the Bush administration. A stem cell is actually sort of a generic term, meaning a cell that hasn't matured yet, that hasn't necessarily decided which way it's going to go and hasn't reached its full capacity. So in the brain, there are neural stem cells, which can become oligodendrocyte precursors and then can become oligodendrocytes. These cells find an axon that's not been myelinated, and they wrap the myelin around it. In MS, however, as I said, the myelin is destroyed in local areas, and the oligodendrocytes that make that myelin are destroyed along with it. Okay, however, there's a conundrum here, and this is the approach that my lab is interested in. There are adult stem cells that remain in the brain. Everybody in this room has them in their brain. These cells theoretically have the ability to become mature oligodendrocytes and make myelin. But in MS, they don't. They might a little bit in early MS, but as MS goes on, they lose the capacity to make new myelin. And we wonder why that is, since those cells are really capable of it. So we ask, what is needed for a stem cell or a precursor cell to make a mature oligodendrocyte and make myelin and remyelinate the MS lesion? So we think about this two different ways. We know that during development, there are factors that promote the oligodendrocytes to make myelin. And maybe those factors are missing in MS. The opposite side is there are also factors that inhibit the development of myelin. Maybe those factors are increased in MS. So the approach we take, so we're trying to figure out what is the approach we can take? How in the world do you find out what these factors are? So we know that during development, children grow and make myelin properly. So if we can study that process, we can figure what makes myelin and what inhibits myelin, what stops myelin. And what controls the generation of myelin are proteins in the brain. And these proteins can either, as I said, promote or inhibit making myelin. So we have been studying a protein in the brain that's very important in the development of the nervous system. It has the unfortunate name of, well, I'm going to call it BMP, but it stands for bone morphogenetic protein. So you're going to think, bone? Why is she talking about bone? Does bone have anything to do with this? No. So I'll tell you a little bit about how science works. BMP is important in many organ systems in the body during development. So a bone researcher first identified it and said, hey, I've got this protein, and it makes the bones grow. It's really important. I'm going to call it bone morphogenetic protein. He found it first, or she found it first. Sometime later, some neuroscientists said, wait, that protein BMP is really important in the development of the nervous system. It's just like that protein that my neighbor down the hall, the guy in the next building, is working on. It's called BMP. I can't give it a different name because that will confuse everybody, but I got there second, so I'm stuck with the name BMP. So that's how it got called BMP. But what my lab has found is that during development, BMP stops the stem cells from becoming oligodendrocytes, just stops them cold in their tracks. So how do we know this? Well, one of the things that my lab does is it can take these cells out of the brain and it can grow them on a petri dish. And we know that under certain circumstances, we can get the cells to make myelin in the petri dish. But if I put BMP on them, it stops them. They will not make myelin. So we and a lot of our colleagues then went into animal models. These would be mouse or chick, or if it's been done in frog too. And we showed that BMP stops oligodendrocytes from maturing. This is all in development. So why would you want a protein in your brain to stop a cell from maturing? During development, wouldn't you want all cells to mature? Well, the way you have to think about it is that BMP is kind of a brake on myelination. So you want your oligodendrocytes to be ready to make myelin when the axons are ready. Your oligodendrocytes are ready too early and there are no axons, they'll die. They need each other, and vice versa. So BMP during development stops the stem cells from becoming oligodendrocytes until exactly when the axons are ready. So what we have shown is that the amount of BMP is very high in development, but around birth, when cells start to myelinate, it goes down naturally. And that allows cells to myelinate, a very nice system that the body has worked out to make everything happen right on schedule. But we wondered about this. We wondered if BMP inhibits myelination in development, maybe it comes back in MS, maybe it's an inhibitor in MS. So we and other labs have looked at this, and we have found that there is too much BMP in the areas of the MS lesions. Way, way, way more than in a perfectly normal axonic myelin. So I'm showing you real science here. I don't want to drive you crazy with it, but this is a quarter of a spinal cord. The myelin has been stained red with a fluorescent marker. The neurons have been stained green with a fluorescent marker. The blue cells on the right are the immune cells that are invading and chewing up the myelin. And we found that in those areas, there's lots of BMP. And we think that BMP is stopping the stem cells from being able to make new myelin and heal the area. To show you some real data, I believe that the animal model probably that you were talking about was EAE. This is the EAE. It's a mouse model of MS. You can see there's BMP. That again, we have stained the BMP with a fluorescent tag, and you can see it there. And probably more importantly is this figure. So when we use our mouse model, we can score the animals by their symptoms, and we find that animals with the worst symptoms have the most BMP. So what can we do about excess BMP? Well, there's a protein in the brain, another one with a funny name. This one is called noggin, like your noggin. That is really how it was named, and not by me. So noggin is an inhibitor of BMP. So if we added noggin during MS, could we stop the BMP and regenerate the myelin? Well, there are some studies, including one from our own lab now, that show if you add noggin to the central nervous system or to cells from the central nervous system, you can stop the effects of BMP. The noggin essentially sucks up the BMP and keeps it away from the damaged areas. So this is what's going on in my lab now. The two projects, the two approaches that we're taking currently, one, since we are a basic biology lab, we don't feel that it's good enough to just know what BMP does. We want to know why is BMP upregulated? Why BMP? Why not one of the other proteins in the nervous system? And we think we have a candidate. We think it's due to oxidative stress in the system. We also want to know if BMP is really the right target. So if we stop the action of BMP, can we actually get enough repair to increase mobility and quality of life? That's a really important question. So we are testing animal models of MS to figure this out. So what I have here is a little bit of a chart, because I got asked this question before, and I think it's important to tell you all how one would take this experiment, and I'm using this as an example, from what's called bench to bedside. My lab bench to your bedside. So the first phase would be understanding the biology, and we're trying to complete that. We think we know what BMP does. The second would be finding therapeutic targets. Well, we've got noggin, but there are other BMP inhibitors, and maybe they work better. We have to try them out. The third would be testing appropriate animal models. To me, this is key. If we stop BMP, will it really be good enough to restore function and to improve lives? What's the best way to deliver BMP? And is BMP all bad? We're working on this, but we don't know the answers yet. If this all works, one would move from animal models to patients, and we would partner with clinicians like Ellen here, and we would try to figure out the best way to test BMP in human MS. Perhaps BMP could be used as a biomarker to show the level of MS by just a blood test, let's say. And then if this all works, one could do clinical trials with BMP inhibitors, and then one would have to partner with pharmaceutical companies. So this is my dream, actually. And we're working on 1, 2, and 3, and I hope we can get to 4 and 5. So what I've told you is how our study of how nerves are protected as they develop has led us to identify a candidate and some candidate proteins that we hope we can manipulate to improve therapy in MS. It's also an example of how basic research in the laboratory can lead to improvement in clinical applications. So we couldn't do this without your help, and I need to throw this in here. Some of you know that research funding from the government has become increasingly hard to get. The headline up there that I can sort of read from here, NIH grant success rate likely hit historic low in 2011, was from Science Magazine a month ago. But thanks to the events that you sponsored, the things that Larry Kane talked about, this research and the research of my colleagues will move forward. So we are very grateful for your support. So I got one more slide. So if I was giving a research talk to my colleagues, which I actually did yesterday at the Society for Neuroscience meetings in Washington, I would thank the people who helped me with this research. This is absolutely standard, and one doesn't want to go without it. So I thought it might be interesting to you all to see how many people go into making a basic research project, because it's all about training for the future, too. So Mary Reed is my technician, and she runs the lab. Stacy Lee, underneath her, is an undergraduate at University of Pennsylvania who is learning how to be a scientist. First steps to learning how to be a scientist in my lab. She comes 3 times a week, and we have her doing some projects. Jill C. and Keith Feigenson are graduate students who trained in my lab. Well, no, not graduate students anymore. They both have their degrees. But they did their training in my lab. Jill worked on the BMP project. Keith worked on a related project, and Keith was actually partially funded by the Delaware Valley MS Society. They now have their PhDs, and they're going on to start their research labs. Jahan Ara did the EAE project. She was a postdoctoral fellow, that's after PhD, and she now has her own lab at Drexel. Heather French is a neonatologist who worked in my lab. Neonatologist, what's that have to do with MS? Well, she was working on a related project on oxidative stress, and we did a lot of cross-pollination and cross-fertilization, and we shared ideas, and she learned from me, and I learned from her. And then I have colleagues and collaborators who I couldn't do without. Steve Scherer is another myelin researcher at University of Pennsylvania. He's sort of my collaborator over at Penn. Becky Simmons is a neonatologist. Again, I'm working with her on a related project. It'll be related to MS. Jeff Golden is in the lab next door. We share students, reagents, and the all-important lab meeting where we bounce each other's ideas off of each other. And then everybody has a research mentor. And mine is a guy named David Pleasure, who hired me as a postdoctoral fellow many years ago. And Dave was Chief Scientific Officer of Children's Hospital until 6 years ago when he headed out west to California to head a new research institute at Shriners Hospital, UC Davis. But since he trained me, he will always be my research mentor. And I actually talked to him today about some MS-related things. So that's the crew. Thank you very much.
All right, so let's have our third speaker, Dr. Simon Gregory, come up, and we'll hear about what we can do in the realm of prevention or the genetics of this disease, and see if we can prevent people from actually developing it.
Number 22. Someone's car's pulled up to the front and I'm calling out the number for the valet. I'd like to thank you for the invitation to come and talk to you. If it wasn't for Tammy, I wouldn't be here, honestly. Tammy and I met probably 3 or 4 years ago when I gave a very early talk about the work, and I'm going to tell you briefly at the end of my time with you. And at that point, we just recently published the finding of probably the first gene associated with MS for 20 or 30 years. And I can remember at the time saying it really was just going to be the sort of breaking of the dam wall. With this one gene and the techniques we were using back then, it really signified sort of the end of one era where we used an approach to identify genes that make individuals susceptible to MS, to the technologies that are now being used to identify literally dozens of genes. There was a publication which I'll allude to at the end of the talk. And one thing I'm not going to talk about is actually yet another technique. We're able to sequence individuals at much greater depth than we ever have before. And that approach will again identify yet more genes and more parts of your DNA which make you susceptible to developing MS. So what I thought I'd start with is just talking about genes, genomics, DNA, and exactly what it is. So you hear genomics quite often in the news, and actually somewhat Interestingly, my number plate is actually genomics, which people think I'm usually a bit of a poser driving around the car that says what it is that I'm working on. But it's effectively the study of an organism's entire genome. And the field includes efforts to identify differences in DNA, not only between organisms, but between individuals who are susceptible to a particular disease. But what is deoxyribonucleic acid, or DNA? So for some of you who will have the handouts already, you'll see what's coming. So DNA is alternatively named for a clothing store, a brand of high-definition televisions. I assume that's perfume, not aftershave, by the bottles. Staircases, earrings, furniture, they're a crystal structure, a tie. I've actually got that tie. And the two guys who identified the double helix structure of DNA in 1953 can actually have bobbleheads of Jim Watson and Francis Crick. So DNA, just those three simple letters, have sort of joined our lexicon of common conversation. And I think as the technologies improve and our ability to generate a lot of sequence, and each of our cells contain a lot of sequence, we're able to generate it at a fraction of the cost. So I spent 10 years of my life working on the Human Genome Project. So I was in charge of chromosome 1 for the Human Genome Project. And I wasn't involved with the project for its entirety. It was probably going for 13 or 14 years. So it took us that long and around $500 million to generate the sequence of one individual that we can now literally do in 10 days to 2 weeks for around $10,000. And so that's where the direction that the field is heading. So you imagine if the technologies that I'm going to start off with, where we're sort of groping around in the dark looking at individual candidate genes to see, okay, is this gene implicated in the development of MS or not? By the end of this year, we will literally have thousands of genomes available in the public domain, not only with respect to MS, but lots of other common complex disorders as well. Okay, so what is DNA itself? So each of us has around 50 trillion cells. I probably have 5 trillion more than I had last year, and as we get older, we tend to gain weight and gain cells, and then we get older still, and we tend to lose weight and lose cells. But we have, on average, around 50 trillion cells. And within each of these cells, we have 23 pairs of chromosomes. The chromosomes themselves are the things that are packaged. So it's DNA wrapped very tightly together in different structures that shows you... So we have these sorts of things as your chromosomes. So when you think of chromosomes, you look at a textbook, and we have this sort of classic chromosome structure here. But those chromosomes are actually made up of around 3.2 billion bases of DNA. Within the DNA itself, we have 21,500 genes. When I first started on the Human Genome Project back in 1993, we thought there was around 100,000 genes. And interestingly, we have around the same number of genes as a fruit fly or a worm. But it's how we use these genes. One gene can produce multiple products that obviously differentiates us, or most of us, from things like fruit flies and worms. The thing is, when you have multiple products of genes, it's the products of those genes and the combinations of these genes that produce these millions and millions of proteins. And the proteins themselves are the building blocks that not only go to make up the cells, but go to make up our bodies. So the really cool thing about DNA is the fact that all living organisms are made up of these 4 basic bases, A, C, G, and T up here, right at the top of the screen. So every living thing contains those 4 bases. And from those 4 bases, we have the instructions for producing proteins, as I just described to you. But those proteins then go to make up different cells, and the cells themselves go to make up, cells the lining of the stomach, cardiac cells, neurons, as we just heard about, and in the lung tissue. And then those cells go to make up particular organs, and then those organs make up particular systems within the body. So you can see that there's a lot of information contained within those 3.2 billion bases of cells, and every cell is just made up of 4 different bases. So this is just a bit of sequence which is near and dear to my heart. So if you imagine 3.2 billion bases, random assignments seemingly by looking at this slide. Random assignments of just A, C's, G's and T's. I can tell you by looking at this, can anyone see there's one particular base that's different? Yeah, well spotted. That one there. So that one is important because that is the individual nucleotide that we identified within a gene that we published in 2007 as being more likely to be contained within individuals with MS than people who don't have MS. People who don't have MS more often have a T in that position than a C. Okay, I'll just pause for the brevity of that moment. So there's 3.2 billion bases, and we found that one. I joined Duke, as you can, well, I'm not going to tell you where I'm from because I've got a slide coming up that hopefully you'll recognise. So I joined a grant that was held by Jonathan Haynes at Vanderbilt, and the grants have been going for 15 years, and they'd spent a lot of time collecting MS patients and using traditional genetic approaches to identify genes that put people at risk of developing MS. And so when I joined that group, we were actively collecting MS patients. And the goal of the grant, which had been running for 15 years, was to identify these exact sorts of things, the C instead of the T. Okay, so if you're not quite sure how difficult it is, I'll just try and enforce that upon you. So if you think of the world as being the whole genome, and over here, each of these are our different chromosomes. So we have chromosomes 1 to 22, the largest to smallest, and they're the non-sex chromosomes. And here we have this is a male because we have the X chromosome staying here in a fluorescent probe or a dye. And this is the Y chromosome here, so we can tell that's a male. But if you just focus on chromosome 1, which is obviously the most special chromosome, and if we look at that in the context of a different, zooming in globally, so we've gone from the whole world, you can see where I'm going with the slides. If we look at one particular chromosome, so chromosome 1, for example, has 263 million base pairs, those millions of those A's, C's, G's and T's. If we look at a particular part of the chromosome, we will see banding patterns that cytogeneticists used to use in the old days to figure out where they were on the genome, where we had losses and gains of material. So that's 20,000 base pairs, 20 million, sorry. If we zoom in a particular region of that chromosome, we go further, we identify a particular gene. So we've gone from effectively saying our whole genome is the whole world, the top left here, down to a particular gene, which is a house on Google Maps. And what we're interested in is this gene structure. Now, when we look at DNA sequence, it doesn't appear as like nice discrete boxes. It's just all those letters, all those A's, C's, G's and T's, all in one field together. And these genes actually make up only 2% of the human genome. So only 2% of those A's, C's, G's and T's actually encode for the protein, which together form the cells or form in combinations to form cells or signalling pathways, etc. So so we get to the gene, which is okay, but we really want the sequence within the gene. So if we go from there, if we look in the back garden of my parents' house in Australia, we actually find a canna lily. So it's effectively looking for an individual change in a base, in a flower, in a back garden, in a state, in a country, and in the world, that we try and identify these particular DNA changes from a T to a G. And so that's the sort of task that we've been laden with to try and identify the genetic variants that predispose people to multiple sclerosis. Now, we've already heard by great talks before me that MS is in fact a genetic disease. So when is a disease in fact genetic? So if you look at a family history, so if you've got grandparents, uncles, aunts, cousins, your children have a particular phenotype, particular disease, then it's predictive of the disease being genetic. even when you adjust for like known risk factors which might contribute to that disease. As we heard, twin studies are very, very important, and actually twins that are discordant, one twin has a disease and the other twin doesn't, are really cool for studying at the moment. Because as you imagine, as we heard, you're genetically identical. So you've got 3.2 billion bases the same, and yet one of you has a particular disease and the other one doesn't. And so we look at things like the incidence of disease in identical and non-identical twins to help us determine whether a particular disease is genetic or not. And then we look at things like is there a ratio is higher amongst first-degree relatives. So the first thing we do when we look at common disease is say, all right, is it genetic? And if it is genetic, we can then start collecting families, which is very useful for identifying genes. We've already heard a bit about this already. So the prevalence, as we know, in the US is some 400,000 individuals suffer MS, which I think is probably an underestimate. We've heard about onset, family history, we've heard about female to male ratio, and then your likelihood of developing it if you're a monozygotic twin, if you're an identical twin. So there are mechanisms apart from your DNA sequence which are important. But why study the genetics of MS? Well, the whole idea is just to give us a better understanding of the biology. As we heard, there's tremendous research that's going on in the field about, just attacking the disease from different directions. It's not only genetics, it's not only potential treatments with vitamin D, it's not only myelin repair, and those, the three talks you're going to hear about tonight are only just three very small instances in which we're trying to identify the causes and cures for MS. But for the genetic sake of it, it's to give us an understanding of the underlying biology. Why are we born with genes that may predispose us to developing MS? And if we identify the genes, does that then identify a potential target that we can use for treatment? Or if you are given a particular drug, and I'd be really interested to hear about the vitamin D study, after the 2 or 3 years, are there individuals who respond to vitamin D treatment and individuals who don't? So if you think about, within one family, you inherit lots of traits from your parents, some good and some usually not so bad. Not so good that you usually display when you get older. But you can, like within my own family, I have 2 daughters, and one of my daughters gets hay fever at exactly the same time I do, and the other one's like the wife, doesn't get hay fever at all. And so we are born with some sort of genetic predisposition to respond to drugs, respond to environmental influences. And so not only can genetics play a part in identifying the mechanisms, but once you have a disease and you are treated with Copaxone or whatever, genetics can play a part in who responds to the particular drug or who doesn't. And so genetics also is good, hopefully useful for predicting who might get MS. And then, importantly for us, understanding the course of MS. Why do some people have long remissions, and long relapses, or frequent relapses and remissions, or get worse after a relapse, etc. So as we've heard already, there's multiple factors which influence complex disease. For MS, it's potential pathogens, chemicals, exposure, diet, etc. So these are the potential environmental factors. We have other things besides just the DNA sequence itself. Once your gene is transcribed, there are other mechanisms which I won't go into which can influence the expression of a particular gene. And there's the genetic variation themselves. So there's multiple factors which influence the DNA sequence that we're all born with. As we've heard, and we didn't actually work together on this, it's not a big plug, but it's a nice synergy I see. There's a definite link with vitamin D and MS, cigarette smoking, and there have been these Epstein-Barr virus molecular fingerprints in the brains of individuals with MS. So you can see, independently, we've come up with the same causes, but we don't know why. Okay, genetic disease. So you can you have common complex disorders. And the reason they're complex is they're caused by multiple genes that cause the particular phenotype, which cause the disease. The easier ones to identify are these sort of Mendelian diseases named after Gregor Mendel, he and the peas. And so what you'll have is one individual mutation which will be located in one particular chromosome, which will cause one disease. And so these types of mutations are they're relatively easy to identify relative to the complex disease. And that's because in complex disease or common diseases, there's multiple different, I won't call them mutations because they're not affecting one particular base pair or combination of base pairs in an individual. These are actually polymorphisms. So these are present in everyone in this room. Okay, so what we're trying to identify is why people with MS have a particular variant more frequently. So you notice I didn't say they have this C instead of a T at this position. I said they have it more frequently. And these polymorphisms are just spread throughout the genome. And, you know, the genetic risk factors which make you at risk for diseases like cardiovascular disease, Alzheimer's, Parkinson's, multiple sclerosis, etc. There's a huge list of diseases which are common complex disease caused by polymorphism. So what is a polymorphism? So we have 3.2 billion of these bases packaged in our chromosomes. And what we're trying to identify is these single nucleotides. So an individual base is called a nucleotide. So we have 3.2 billion nucleotides in our genomes. And what we're trying to identify are these polymorphisms. So you inherit one chromosome from your mum and one from your dad, and you may inherit a slightly different DNA sequence in one position in your dad compared to your mum. But the sort of, which sounds sort of, you know, reasonably easy to figure out, but the problem is we have 3 million of these variants within our genomes. And these 3 million variants, so I'm throwing a lot of zeros out here, 3 million, 3.2 billion, etc. These 3 million variants not only make us susceptible to disease, how we respond to particular drugs, but it also defines who we are, what we look like, how tall we are, how short we are, how quickly or not quickly you lose your hair, one the former. But we inherit these genetic risk factors or factors about our development from our parents. And so we try and identify these single nucleotide polymorphisms within individuals with a disease more frequently or less frequently than what we call controls, so matched people of the same age, sex, race, etc. So all we're trying to do is identify, okay, in this mixed population here, the cases are the ones in blue, the controls. So case would be MS in this case. Controls would be someone who doesn't have MS. And we look for the frequency of a particular variant. Remember, we have 3 million of them that are present more frequently in the case population. So in this case, it's the big A than the controls, which have the little a. So as you can probably appreciate, it's like finding a needle in a stack of needles. So not a needle in a haystack, but a needle in a stack of needles, because we have 3 million of them. So I'll just replay that in case all there it goes. I'm sat there trying to see. It took me ages to get the right PowerPoint thing. Right. So there's 3 different approaches that you can use for identifying these variants. So I talked about how important families were. So I might work the other side of the room now. Like a motivational speaker. Come on. So there's 3 different approaches you can use to identify these variants in a population. So you can use linkage, and this is where new faces to look for. So this is where families become important. So I talked about multi-generational families, and as I was talking to the couple at the Golden Circle event before, families are important because what it enables you to do is look for regions of the inherited genomes which segregate with the phenotype. So what that means is regions of the genome which are in common between individuals who have MS. And if you think that every time you inherit chromosomes, they're all jumbled up from your mum and your dad. So your kids will have a jumbled up combination of chromosomes that you inherited from your parents. And all this jumbling will causes mixing up of the DNA, if you like. And so what we want to look for is saying, all right, is there a particular region of the genome which is always separating with people who have MS, and then how do we identify those regions? We use linkage to do that. There's this other approach called a genome-wide association. So instead of looking in families, what this approach basically does is says, all right, I need lots of individuals who have MS, and let's match them against lots of individuals who don't have MS. And there what we want to look for are the particular base differences, but a frequency. So if you think about that C in the red box with all those 3,200 bases that I had on the screen before, we want to see, do the people with MS more frequently have a C at that position than they have a T at that position? So you can do that on an individual basis, which I'll talk about in a second, or you can look across the whole genome at once. And technology has allowed us to look at hundreds of thousands, if not millions, of these different bases at once. They tell a lot of the story, but they don't tell the whole story. But what I want to talk to you about is what we published when I first met Tammy, when we published in Nature Genetics. It's just this candidate gene approach. And there what we did is effectively said, all right, I literally sat down and I've still got it's an Excel spreadsheet, so it's just a list of genes from 6 different studies or 9 different studies. And I very naively color-coded each of the genes that were identified in a different colour and then sorted them all by name and said, all right, which ones were differentially expressed. So by that I mean, if you remember the gene at the bottom, the picture of my parents' house, and you have the gene next to it. From that gene comes mRNA. So your DNA is the is the blueprint. Your mRNA are the instructions from the blueprint that do things in your body. And when we look at mRNA, we look at expression. So the parts of the genome that are expressed. So all I did was combine fantastic pieces of work from other people and said, all right, what is commonly differently expressed in these studies? Whereabouts are the regions of the genome which have previously been linked? And is there a function of these genes that might be important in the etiology of MS? And so it really is just a matter of me lucking out and combining other people's great work to identify a gene. We published, as I said, at Keith going on about a few years ago. So 9 different expression studies, linkage, etc. And we identified this gene called the interleukin-7 receptor. And this is the point at which we could have stopped. We could have said, wow, fantastic, we found a gene, and let others follow it. But the research team at Duke were pretty adamant that we wanted to figure out what the function was. There's no point in just identifying a gene. As we heard before, you can stop at BNP and say, all right, it's present in loads of elevated levels of expression in particular regions of the brain, and just leave it at that. But it's important to understand the mechanism if you can then try and control it and figure out how it's related to the development of MS. So previous studies had shown that it's important in the EAE model that we've already talked about. It's differentially expressed, and previous genetic studies might suggest that it would be important. Now the reason I show this isn't because I want you to take anything home from it, really. It took me ages to draw this slide, so whenever I get the opportunity, I put it in. So if you imagine your T cells, so we've already heard that MS is thought to be an autoimmune disorder in which the T cells of your body may attack the myelin and be the causal factors of developing the disease. And here up in the top right, we've got the protein. So we go from DNA to RNA, and the proteins produced by the RNA. So that's the interleukin-7 protein itself. So what the gene is that we identified was this protein here, so the blue part. So if you imagine your T cell, this is the surface of the cell, and the IL-7R gene is a receptor. So the idea of a receptor is you get a signaling molecule like this, it binds to the receptor and it transmits the signal through the membrane of the T cell and then does a particular function so that signaling pathway should enact. So we identified IL-7R, so the interleukin-7 receptor, and it's a receptor to IL-7. This is the bit that took me ages. I made a stupid slide. It's one of those things you start and you think, yeah, it's a really great idea, and then like 3 hours later and what happens is you have binding of the ligand, as you call it, the IL-7 protein signaling, you have phosphorylation, you have the signaling by JAK-STAT pathway, you don't need to know this. But the important thing is that the signaling actually results in multiple different effects within the T cell. So we're at the point now of saying, all right, is it important for differentiation? So taking a T cell and differentiating into different types. Is it important for cell memory T cells to be maintained in the periphery? Or is it important for proliferation? So the multiplication of these types of cells. We just don't know. And so the really other interesting thing about it is this receptor comes in 2 different forms. Remember I said what makes us complex and makes us slightly or marginally more interesting than fruit flies is that genes are able to produce multiple different types of proteins from the same gene. Well, IL-7R actually produces this receptor version of the protein, but also produces a soluble form of the protein. And the soluble form of the protein actually binds to this protein that triggers the signaling and reduces the signaling by binding it outside the T cell. And so what we found was that, so this is the genetics bit. So I talked about all these single nucleotide polymorphisms. So they all have these RS numbers at the top, so they're sort of naming convention. This is IL-7R gene itself, so we have the RNA or the protein coding bits here. That's all. And then, so down the bottom, it's just a table of how many thousands of individuals with MS we had to test to identify a genetic effect, which is actually pretty small. So it's your risk of developing MS has only increased by 20% by having a C instead of a T, which may sound a lot. In cardiovascular disease, if you smoke, you increase your risk of having developing cardiovascular disease by 400%. Okay, so it's only a little increase in risk, but it's going to be part of the picture that defines what MS is. So, together with collaborators from the UK and in the US, who were fantastic in contributing their samples, we literally needed thousands of MS patients and controls and family members, etc., to identify statistical significance of this blue SNP up here, so the single nucleotide polymorphism. So that's the C SNP, if you like, that I talked about. And what we found is that individuals who have that particular C produce more of the soluble form of the protein, so decrease in signaling that way than having the T allele. So I just want to talk quickly about genome-wide association. So, well, before I talk about that, so critically, the National MS Society provided funds for us to follow up on IL-7R. As I said, we could have left it and we could have just said, all right, we've identified genetic association, but the National MS Society stepped up and funded, we're probably halfway through that grant, to identify what the particular mechanism is. I'm working with other investigators at Duke to figure out why it produces more of the soluble form than the receptor form of the protein, and we've actually, in the last couple of weeks, identified some really interesting proteins, other proteins which regulate our gene. I have an Italian postdoc who is recruiting MS patients, and in these MS patients, we're saying, all right, can we find the patients who have 2 Cs, remember we have a C from your mum and a C from your dad, 2 Cs and 2 Ts, and see whether there's anything about their T cell function. And I'm glad she's finally got the conditions for the flow sorting of T cells sorted out, because we've been bleeding ourselves to get the conditions right. It sounds pretty gruesome, but it's fine. So you literally take blood from an MS patient and you sort the particular cellular components of your blood, and we're interested just in the T cells. But we're starting to think about an animal model. So we've heard a couple of times already tonight about how important animal models are. Now, interestingly, the mouse doesn't produce the soluble form of the protein. So what we want to do is introduce the soluble form to the mouse in a strain that's susceptible to autoimmune disease, and see if we can trigger something other than an EAE type experimental autoimmune encephalomyelitis, which we've heard about. An EAE mouse model of MS to see if we can understand the mechanism better. Okay, so I just want to finish briefly, and probably this is the most exciting piece of work that I haven't been involved with, which came out this year. And so what you do is, instead of looking at individual SNPs across the genome, as I said, you can look at millions of them. So it's an unbiased screen. You're not saying, all right, I just want to look at my candidate gene like I did with my different expression studies and linkage and some other genetic association. You're just saying, take the whole genome all at once. Take these millions of SNPs and saying, all right, can I identify genes which are, or these SNPs which are more frequent in MS patients versus people who don't. And this year, so when I was involved with the International MS Consortium, there were 4 groups. That expanded since our discovery in 2007. There's now 20 groups. And so Stephen Saucer and colleagues from the UK, Steve Hauser at UCSF, David Hafler at Broad, and many groups around the world. And you can see here, so all the red numbers are MS cases, and the black numbers are controls. And below that, you've got a really interesting population stratification. So that means that people from different countries are different in their genetic component, as you'd expect from people from other countries. But what they did was identify all these genes, which are statistically significant as being associated with MS. And so I obviously put a red box around the most important one there. But this is what I alluded to when I started this talk, and I first talked with Tammy all those years ago, is that we had just identified this, and this has all happened in 4 years. And remember, we identified ours, and nothing had happened for 20 years before that. So genetic studies are really progressing at light speed. The next bit is to understand why something like IL-7R or all these other genes are actually important for the development of MS. And to do that, it really does need research funds. As we've heard, the funding from the NIH is absolutely appalling. Now, luckily, my work is funded for another few years, but for young investigators or those who are starting to explore these genes, it's really, really dire. Of all the grants that are submitted to the NIH, probably 50% are scored. And if you think there's thousands of grants going in, of the 50% that are scored, 10% are probably funded. So you're looking at less than 5% of the grants that are submitted to the NIH are funded. And it's not to say the other 95% of the work that's being submitted isn't important. It's all important. But that's where the foundations like the National MS Society are critical. And as Tammy alluded to, it's really, really dire economic times as well. So scientists like myself and the other 2 speakers before me are just sort of, it's a double whammy where it's really tough to get science funded at the moment. And so the work that you do with the mud run, the walks, the bikes, etc., absolutely critical. Absolutely, if we're going to continue this research. That's a fairly gloomy note. So I've got loads of collaborators from lots of different places. I'll just highlight the ones that are still, we've continued this work on to figure out what IL-7R does. So Mariana Gutt, it's funny, the NIH grant that I have and the MS Society grant includes myself as an Australian, Mariana is Puerto Rican, Jorge Oxenberg, who works with Steve Hauser, is from Argentina, Irina's Russian, I've got an Italian postdoc, but thankfully Lisa Barcelos at Berkeley's American. So it's a bit like the UN when we're on our conference calls. But anyway, with that, I'll thank you very much.
I'm going to invite the speakers up, and we'll open the floor to some questions. So if you just have a question, just raise your hand, and somebody will hopefully show up in your row with a microphone. So we got something over here.
A question for Dr. Maury about the vitamin D study. It said that it would be done with Copaxone. So will they do a study just by itself? Can it be done by itself?
That's a great question. So why did we decide that all the patients in our vitamin D study would be treated with Copaxone? The reason is that I don't feel in this day and age that it's ethical to do a study in people with relapsing forms of MS without giving them a medicine that we know works. So for me, it would be hard for me to ethically enroll a patient into such a study, and therefore I really felt like it was important that we had a platform therapy available to all of our patients. And we wanted everybody to be on the same one so that it didn't create other noise in the study, so that what we were looking at was a pure effect of high or low dose vitamin D. And for a number of reasons, we chose Copaxone to be that base medication. I think in the future, if you were to show that high-dose vitamin D was effective, you could then consider doing it as a head-to-head study of high-dose vitamin D versus another medication. But I think right now, until we know that vitamin D is helpful, it would be really unfortunate to have people in the study without a medicine that we know works.
I have one over here.
Is there any age that you shouldn't take vitamin D?
So is there any age that you shouldn't take vitamin D? Well, we certainly know that the vitamin D requirements change as people age. So older individuals just have lower vitamin D levels in their blood than younger individuals. And as such, the recommended daily allowance for vitamin D is higher for those who are older than 70 than those who are 10. But what the actual doses are that people need in health and then in disease, we don't really know yet. Jeff.
What are the target blood ranges for vitamin D that you're looking at in the study? I didn't quite catch that.
Okay, so we're trying to target, we chose the 5,000 international unit daily dose because we think that it will land most people in what I think is the best supported target range right now, which is between 40 to 60 nanograms per milliliter, or 100 to 150 nanomoles per liter. That's how it's measured in your lab.
Hi, this message is for Dr. Greenspan. You stated that children or babies do not have myelin sheaths and they only develop it as time goes on until they're, I guess when they're 25, that was the age range that you gave. What about people that develop symptoms for MS prior to the age of 25?
Well, they have some myelin. I mean, I'm not a clinician, so I can't tell you the statistics, but most myelin develops in the first couple of years. It's just, I threw out the statistic because myelination isn't complete basically until early 20s. So there is considerable myelin in a young person, so that can get attacked in pediatric MS.
So just to add to that, when you look at most babies myelinate within the first couple of years, and then it slowly evolves over time. And interestingly enough, there have been some studies recently that looked at the frontal lobes of teenagers. And the frontal lobes of teenagers don't myelinate really until they get into their 20s. And people believe that is the explanation for teenager.
I have a question regarding primary progressive MS. It's not being included in the vitamin D study, and then I don't know about the Dr. Greenspan's talk about myelin. If you see differences or you're looking for differences between relapsing and primary progressive. But primary progressive isn't getting a lot of press here.
So I'll start. I am actually really interested in understanding if vitamin D is important for people with progressive MS. To sort of break it down in an uncomplicated manner, many of us believe that progressive types of MS are related to the damage to underlying nerves, like Dr. Greenspan was talking about, rather than due to inflammation causing the coating of the nerves to come undone temporarily. And so we think relapses are often because of the coating of the nerves coming undone. And that's what we're sort of studying in the clinical trial, relapses and spots on the MRI scan. And since those outcomes don't really occur in progressive types of MS, we think, we can't really include people in a study in which that's the endpoint. But I am working actually as part of my NIH-funded grant to look at, in general, whether vitamin D levels are associated with loss of nerves themselves. And I think that it's possible that it actually is important in protecting nerves, because we know that nerves have receptors for vitamin D, and so vitamin D is acting on the nerves themselves as well. So I think that will be a really exciting area of research, and I hope a very promising one for people with progressive types of MS. And in the clinic, I do talk about vitamin D supplementation with my patients with progressive types of MS.
Okay, so in answer to your question about my work in different types of MS, we haven't really looked at that yet, although it's something that we would like to look into. There are ways to manipulate animal models to distinguish between the different types of MS, depending on how you set them up. And that is something, it's a great idea and something we want to look at in the future.
You know, one of the biggest challenges we have in the field these days is that, you know, all the studies are geared toward relapsing, remitting, and not enough toward any progressive, whether it be secondary or primary progressive. And that mainly comes out of the fact that we just don't know how to study progressive disease very well. We don't have good outcome measures. It takes a really long time for progressive disease to change that's measurable over short. Like if you try to do a study over 2 or 3 years, you're not going to see a huge change because it's very slow. So that's been the challenge that we have, that we don't have sensitive metrics to really assess that. But we're working on surrogate markers, whether it be MRI or other things that may be helpful in that regard.
I just want to say something because no one's asked me questions. So in the genetics field, and I've been asked this question before when we talk about the studies we do in genetics and why are they most relevant? Well, why do they focus on relapsing remitting? And part of it is because of the numbers. So if you think that 80 to 85% of MS cases are relapsing remitting, and to do our genetic studies, we need literally, as you saw, thousands of cases to be able to identify these small differences that are present within everyone. And it's really, really tough to get, large cohorts of primary progressive MS patients. And they're an underserved part of the MS population.
Somebody down there?
Yes, hi. On that note, I have relapsing remitting MS, but I haven't had an episode in many, many years. And this past winter, I was very sick. I didn't contact my neurologist. I went to a new MD who did so many blood tests that they had to give me orange juice and donuts after all the blood that was taken. And they determined that I had significantly low levels of vitamin D. And I all those symptoms I was having, I thought I either had cancer or like intestinal cancer, or I was having heart problems because I was in a lot of pain every day. And as soon as I started taking 5,000 IUs, maybe it was from your research, but as soon as I started taking it, within a few weeks' time, I started feeling better. But because I am an animal rights advocate, I didn't know that vitamin D3 came from lanolin, so I switched to the ones derived from mushrooms instead, but I still feel really good. And I'm going to have another blood test in a few weeks to test to see if my levels have come up. But I didn't even want to start taking vitamin D until my neurologist told me how severely, he said I was profoundly deficient in vitamin D. So I just thought I would put that out because it's really working. Thank you.
Great, thanks. Back over on this side. Back over there.
Kind of a follow-up question to that, which is if someone is, with all the studies for vitamin D that are out there and giving different levels of what should or can be taken, for a lot of people, for a lot of us with MS, we have more than one doctor that we see, a neurologist or an internist or optometrist. And they give you different answers for different things. So how do we determine what the right level of vitamin D is? Do you know, is it 1,000? Is it 4,000? Is it 5,000?
Well, part of it will be dealt with in this study. We're hopeful, but why don't you go ahead and give your opinion?
Well, so even though I told you I don't think we have fabulous evidence to not do a clinical trial of vitamin D, in my practice I do talk with my patients about vitamin D, and I say we can measure your level, your blood level, and if it's lower than that range we think could be helpful, I can give you vitamin D, and we can recheck the level after you've been taking it and make sure you end up in that range. And we might be making a mistake because maybe, like some of the other supplements, it's actually harmful. But the evidence is supporting more of a helpful than a harmful benefit to risk ratio at this point. And so I always have that discussion with people rather than sort of giving it in a sort of a blanket form. And then I basically suggest a dose that I think will get them into that range, and we just follow it up as in the other woman's discussion there in a few months, and if it's not there, we increase the dose until we get to that range.
And in some cases, some physicians believe just a standard 2,000 units or something 2,000 to 4,000 units is not a bad range to take anyway, even though we don't have much data to support it.
It's good for seasonal effectiveness.
Back in that corner.
Yes, my question is for Dr. Gregory.
All right.
I like the question already.
Forgive me if you mentioned this in your presentation, but you lost me at Australia. For someone with MS who is considering starting a family, how accurate is the genetic testing that's available to people in determining the risk of passing MS onto their offspring?
So it's a very good question. It's the first question I always get asked. You know, at the time we'd identified, in addition to the MHC, we'd identified IL-7R as a risk factor for developing it. Now we have 57 other genes. And when they did sort of the gene-by-gene analysis of these 57, it wasn't really greatly predictive of whether you would, you know, do you need all 57, do you need 20, do you need 10, whatever. It's not really accurate at predicting, you know, the likelihood that you'll develop MS, because you have to remember that, you know, it's a complex disease, so it increases your risk of developing the disease, but there's absolutely no guarantee. If you think back to the myozogoic twins, where only 20 or 30% go on to develop it, and they're identical, and still not 100% penetrant, as in not if one twin develops it, the other one will as well. Now, it's a tough call, but at the moment, genetics can't give you any clearer information as to whether your children will develop the disease or not. it's not like a monogenic disorder where they actually do prenatal screening for very severe monogenic disorders, and because it is highly penetrant, if you do inherit that mutation, you have a very, very high likelihood of developing it. What I usually end up finishing to say is that you just have to watch out in a family that's predisposed. So, assuming that you're not the only person in your family, but you have a family history of developing the disease, it's just being more vigilant. So, whether you need screening early on, whether you don't just treat CIS as an isolated cause of some inflammation rather than it being a sentinel event which may lead to MS. It's just being more vigilant in your children, I think. And then, to seek out the good neurologists that you have yourself for your own disease. Talk to them about your kids and just watch your kids. But it's very tough because it's not predictive.
So I have a add to that. So a lot of patients will come into the office and say, I'm interested in having a family. And they're worried that they're going to pass this disease on to their kids. So we tell them all sorts of things. But I'm curious as to what your thoughts about that conversation should be.
So
I came from Australia and I know all about it.
Yeah, I've had a wine already as well. So I gave neurology grand rounds at Duke, and it was soon after we published, and there was a neurologist who stood at the back and really was adamant that what we found had no significance to his patients, because we could test you for it, but it's still an increased frequency of this variant over the normal population. And he was saying, what is the point of doing this research until there's point of care where you can actually affect some sort of change in a patient's outcome? Does it influence the way a drug's metabolized or whatever? I think the thing to say would be just to make them aware of the fact that it is a genetic risk that has environmental exposure which may trigger the phenotype. And your risk, even if you look at all of those, the MHC is just hugely significant, 1501 variants. And yet we still don't know how that works. Each of the variants that we identify at most increases your risk by 30%. And so you can't say you have this variant, you will develop the disease. You have to think about all the environmental exposures that the individuals are being exposed to on top of a genetic risk. And even if you had all of the variants that were published this year, you may never go on and develop the disease.
I think that's really important to keep sight of, that you're looking at a couple of percentage point risk for a child, a parent who has MS, 2-3%. So we keep it in the correct context.